So today I have the pleasure of talking to Beverly Mok. She is a chemistry and chemical biology graduate student at Harvard University and a member of the Liu Lab at MIT and Harvard Broad Institute. Now, she did her undergraduate studies at Cambridge University in England. That's correct, right?
Yeah, that's right.
So suffice to say, she's an accomplished young scientist. So whenever I do this, Beverly, I try to give my guests a chance to give us their journey through science, right? So
Right.
in your case, if you can at least just let us know how it all started for you, right? So where did you do your undergrad and how did you actually get to that institution? And how did you make your way to Harvard, then to the Broad Institute? And according to one of our email correspondents, right, you did
Right.
say you're from Singapore. So
That's right.
I'm interested to know, how does a Singaporean find a way from the beautiful country of Singapore to you know Cambridge, Massachusetts?
Right. Yeah. So like I said, right, so I grew up in Singapore. I spent like the first 18 years of my life in Singapore, like going to like middle school and high school. And I think growing up, I was influenced by my older sister. She was always into the sciences, and right now she's a chemist by training. So growing up, I was always more in tune, like to the sciences, right? And in high school, they had after school attachments where they bring you to like local universities to try your hands at some wet lab experiences. So, I mean, I played around in some labs that did like organic synthesis and catalysis, but it was never really something like I never really thought I could make a career out of it. So when it came to undergraduate, like you had to apply to university, it was more like a narrowing of choices rather than what I wanted. So it's like, okay, finance was out. I didn't like any of it. Engineering, nah, I wasn't like really good at math. So after whittling everything down, I was like, okay, maybe I could do like pretty okay in chemistry. And the Singapore government actually has this program where you can apply for a kind of scholarship, and if successful, they will fund your undergraduate and graduate studies. So it's a kind of commitment that you make to them when you're 18 years old and say, okay, I'm going to do science for the next 8 years of my life, get a graduate degree, and return to Singapore and work at a like government research institute. So I was naive, and at 17 years old, 17, 18 years old, I was like, okay, I'm going to do it. So then I signed with the government agency, and they gave you the opportunity to pursue a education overseas. So my first thought was, looking at the universities in both the US and the UK. And at that time, I had a few of my seniors from the same high school as me who were already in Cambridge University in the UK, and they had many good things to say about the science program. That was how I applied to the university and got accepted. And in the UK, the undergraduate program is a 3-year program. And the big difference I feel compared to the US is that in the UK there is no term time research, meaning it was just all like book work essentially. Right? I mean, you had practicals, but then they were part of the curriculum. It wasn't more like you had to take a semester out to do like research in a lab, like how is it in the US. So I guess my first, like the turning point in my undergraduate degree where I really knew that research is something I wanted to pursue even further was when in my second year there was a call for applications to a research program at Cold Spring Harbor in the US. So I applied to that, and I was attached to a cancer lab within Cold Spring Harbor. It was run by Professor Chris Kosh, and that lab studied like how the like certain what sort of like processes affect the leukemia progression, and it was a cancer biology lab. And coming from a chemistry background, being exposed to like this different areas of research which I've never had a chance to explore before, like really opened my eyes to like thinking about how I can maybe consider a career that interfaces between like chemistry and biology and medicine. So so that experience was great. It was a 2-month attachment, and following my my graduation from Cambridge, it was a time to apply for grad school. And at that time, before applying to universities, I've come across papers from the Liu Lab in Harvard. At that time, I think it was in 2014, 2015. So it was when genome engineering was like starting to take off, but it wasn't as yet as mature as it is right now. So the main papers from that lab were mainly like in DNA template synthesis and protein evolution, like directed protein evolution. It was a very like tech dev lab, and like just reading the papers just like blew me off, like the kind of ideas that lab came up with. So I think I applied to Harvard mainly because of David Liu's lab. I mean, I applied to other schools as well, but when the offer came, I was like, okay, I was pretty set on going to Harvard. It's like to a different Cambridge this time around. So yeah, that's how I guess that's a brief summary of how I ended up
Wow,
where I am right now.
that's awesome. Wow, that's great. So you did talk about gene editing, right? And that's
Right,
actually the core of our conversation today. Okay. So
right,
so from the very first time I heard about CRISPR, which
right,
stands for clusters of regularly interspaced short palindromic repeats. Am I correct?
right.
Yeah.
I think you're right.
Yeah.
It's a mouthful.
Yeah, I was totally enamored by the technology, right?
Right,
So now, for those who don't know, CRISPR functions as an innate immune system for bacteria and other microorganisms, right?
right,
So now these pathogens are actually themselves infected by viruses, so they can actually get viral infections, and these viruses are called bacteriophages, right?
right,
So bacteria have developed this mechanism whereby they keep an RNA copy from a previous bacteriophage attack, just in case
right,
of a future attack from the same virus, right?
right.
So they can then use this as a guide, right?
That's right.
And they compare it to what's commensurate, an RNA strand or a DNA strand that's commensurate to whatever they actually have in their system, right?
That's right.
Yeah. So at this point, they can then use some form of molecular shear. So in some
Mm,
cases, it could be Cas9, Cas13,
mm,
and they then snip the RNA and DNA of the bacteriophage,
that's right.
and this then renders the attack useless, right? So we've now been able to jerry rig this mechanism, right, and use it to basically edit genes of other organisms, right? And
Right,
as a matter of fact, we can use it to edit genes of human beings, right?
right.
Now, there are a lot of, I'd say, ethical questions that arise when it comes to gene editing, and I hope it's something we can touch on at some point.
Right,
But
right.
I'm interested to know how and when did you actually come in contact with this technology, and were you as exuberant about it as I was when you first heard it?
Yeah, so I came in, I first heard of this technology when I was doing my attachment at Cold Spring Harbor.
So,
So, yeah, so that was probably maybe I was like 20, 21 years old.
mhm,
And at that time, I had an inkling that it was useful, right? Like it was pretty cool, but I don't think I had the scope of knowledge to really realize how groundbreaking this technology was and how was it going to revolutionize the way like a lot of genomics research is being done.
sure.
And it was only after I started grad school and in David's lab when you start talking to people who are actually working on projects that use these tools, right? People that try to use these tools to create new therapies or use these tools to do large scale screens to identify like disease targets, that you actually realize how important and useful it was.
Yeah.
Yeah,
Wow, interesting. So you're in the Liu Lab, and that's actually a lab in the Broad Institute, right?
that's right.
A lot of people don't seem to know that place at all, that academic institution at all, right?
Right, right.
So it's some sort of alliance between two, academic juggernauts, MIT and Harvard,
That's
that
right.
some people would think they have, like a, friendly adversarial, relationship.
Right.
So, how did you first hear about Broad? And there's actually a superstar at Broad. His name is Professor Feng Zheng, right? And I'm
Right.
actually, totally enamored with that gentleman. And, I've watched a bunch of his videos and what have you. And I'm just curious to know if you've ever met him before. But if you can start off with how you found out about Broad.
Right. Yes. So I joined Broad because my advisor, like David Liu, moved his physical lab space from the Harvard chemistry department to the Broad. Right. So that happened in my first year of graduate school. So I knew of the existence of Broad, but then it wasn't like a choice that I could make, like, oh, I want to work at the Broad. Right. I mean, if there are professors that I'm interested in at the Broad, I could apply to work for them. But because I was with a professor that actually had his initial lab space in Harvard, and it was because he moved, and that's how I ended up at the Broad. And as to whether I've met Professor Feng Zhang, I've not physically like introduced myself to him, but we have a lot of events, and sometimes like obviously he gives a lot of talks and presentations. So I've like heard him like speak before, and I've interacted with some of the members from his lab. But I hope sometime in the future I would have a chance to like talk
Right.
to him.
Yeah, and let him know he's got a fan in me.
Yeah, I'll let him know.
All right, that's cool. So I stumbled upon your work, right,
Right.
and online, and I knew right away I had to talk to you about it, right? So now,
Right.
get us going. Now, why haven't we been able to edit mitochondrial DNA up until this point, right? So CRISPR has been with us for some time now, and why has DNA editing in the cell's powerhouse, so the organelle
Mm.
mitochondria, why has it been so elusive to us, right? And I mean, again, we've done editing in DNA, so the nucleus of the DNA of the cell. So how come this is something that we're just starting to get our or wrap our heads around?
Right. Yeah. So yeah, this is a really good question. So if we backtrack a little bit and trace the origins of the mitochondria, like how did this powerhouse organelle came about? Like there's this theory that it originated or evolved from bacteria, like
Right.
bacteria.
Is that why it's so similar? Like its
That's
structural
right.
makeup is so similar to bacteria?
Yeah, that's right. Like as you mentioned, you have like the double membranes. You have the inner mitochondrial membrane and the outer mitochondrial membrane, and it has its own like DNA.
Okay.
And the
That's
DNA.
interesting. So that's kind of analogous to is it gram negative bacteria?
That's right.
They have a double sort of membrane, right?
Right. Right. Yeah, and then it has its own copy, its own genetic material, which we call the mitochondrial DNA,
Sure.
and there are like multiple copies of it. Right. So within a mitochondria, you can have multiple copies of mitochondrial DNA, and each of them could be slightly different in sequence compared to the other. But to answer your question, I think it has to do with the double membrane that we see. So I think as of now, there hasn't been any reliable method or established pathways to import nucleic acids, or in this case, RNAs, into the mitochondria. So given that CRISPR-Cas9 relies on an RNA molecule, the guide RNA, to direct it to where it goes on to its target site, if we cannot import that targeting guide or RNA into the mitochondria, then we're going to have troubles using CRISPR-Cas9 to edit the mitochondrial DNA.
Interesting, interesting. So this process of mitochondrial DNA editing is based on utilizing certain toxins, right, produced
Right.
by bacteria. So more specifically, that would be the Burkholderia cenocepacia bacteria, right?
Right.
When I was actually looking into it, is a comorbidity for cystic fibrosis, right?
Yeah, yeah, that's right.
Interesting how, such a pathogenic organism could actually, create molecules that could actually save life, right?
Right,
All right. Now, can you talk about how the components of this toxin are, I guess, separated and
right.
used in the base pair location process, and then combined, I guess, to cleave base pairs, right?
Right.
How does that work?
Okay, so I'll have to give all this credit for deaminase to our collaborators. So it's a Professor Joseph Mogus at University of Washington and his amazing postdoc Marcos de Moraes. So they were the ones who identified this deaminase
Sure.
from the Bacteria Sinosephacia pathogen, and they solved the crystal structure of the deaminase, right? And it's a relatively small protein. It's only 138 amino acids. And with the crystal structure, we decided that we needed to split this deaminase. And the reason being, if we just transport the entire intact deaminase into a cell, it's going to be really toxic, because you can imagine you have this very reactive warhead just going around deaminating like any cytosine bases that it sees in double-stranded DNA. That's like unleashing trouble onto the cell, right? So the idea for separating was therefore proposed, because when you physically separate you basically like split the protein to two halves. Each half would be inactive until it finds its target site and reassemble to form the fully active deaminase.
Wow.
Right. And so to go about splitting the protein, we relied a lot on the crystal structure. And because from the crystal structure, we could identify regions in the protein where it wasn't very structured. So those are mainly the loop regions. And the idea was, if you could target these loop regions, you could potentially split the protein without causing much perturbations to the overall protein structure.
Sure.
Right. So now we've got a case where we've got two inactive protein halves, and then how do we bring them to search for its target site? Right. So then that's where the DNA binding proteins come in. So each half, or each deaminase half, will be fused to a DNA binding domain. And that DNA binding domain is responsible for interrogating the mitochondrial DNA to search for where it should land on. And if we have two DNA binding domains that are flanking a target site, and both of them land on the target site, land on the flanking regions, the two deaminase halves can then come into contact with each other, because they're in close proximity, to reconstitute the deaminase activity to modify DNA.
Wow.
Yeah.
Wow, that is mind blowing. Wow. So deaminase, that's basically the process of removing amino groups from molecules, right?
That's right.
Wow, interesting. That's just fascinating stuff. So this is one of those podcasts that I listen to like several times.
I mean, I'm always still learning new things too every day. So
It's awesome. So this toxin, is this just specific to Sinosephacia, or is it I guess native to other bacteria pathogens as well?
yeah, I think this it wouldn't be the exact same deaminase,
Oh,
but it is proposed that there are other like pathogens that could have a similar kind of toxin. They could be deaminases as well, but then the properties might be different. So, like our collaborators are actively following up on any novel or previously uncharacterized deaminases to see what sort of other interesting properties they might have that could be exploited for different applications.
all right. So that brings to my mind, how did we find this? I guess this characteristic in the first place, right? The
Right.
fact that this is such a useful molecule that we can use for mitochondrial DNA editing, and how can we use that same process that we used to find this, I guess, regime,
Mm.
and other pathogens that have that same molecule?
Right. Yes. So there was earlier work published by a group at the NIH in 2011 where they used a lot of sequencing approaches, and so they identified they did like they sequenced a lot of different bacteria, like the genomes of many different bacteria, right? And once you've gotten their genomes, they could do alignments to uh, sequences of deaminases that we already know, and see if is there any homology between what is already known out there and uh, what could or any new deaminases that could be found in this bacteria. And they did that as well as a lot of like structural homology studies. And that group at NIH found that there's actually this vast reservoir of proteins that are predicted to be deaminases but have not yet been characterized.
Interesting.
Yeah, and I think, and our collaborator had the insight to focus on this one particular strain of bacteria and performed more in-depth characterization studies of this deaminase.
Wow, interesting. So I guess the end goal is to solve a lot of physiological problems, right, that are
Right,
caused by deleterious genes. So,
right.
just in case anyone is wondering, what ailments do we know that are caused by mitochondrial DNA mutations, right?
Right.
So I know, I mean, I'm not too sure about
Mm.
that, but there's some speculation that autism can actually be traced down to mutations in mitochondrial DNA. So what do we know about all this?
Yeah. Okay. I wouldn't be sure about autism. I'm not familiar with the aspect of literature, but the more commonly associated diseases are usually um, they manifest symptoms such as like neuromuscular degeneration, like hearing loss, muscle weakness, or vision loss. So there's the more well studied mitochondrial disease is this. It's the short form of it is called MELAS, like MELAS, and MERF syndrome. So, I, like these patients, they can exhibit symptoms like really early on, like say when they are 2 years old. And some of the symptoms would be, like as I described, difficult muscle weakness. They have like progressive like vision and hearing loss. Sometimes they have like seizures and epileptic fits. And many of them actually don't live that long. Like it's a long lifespan is not expected. And I think one interesting thing to note about many of these mitochondrial diseases is that the mutations in your mitochondrial DNA can usually either inherited from the mother, so it's maternally inherited, because that's how the inheritance of the mitochondrial DNA is like. It's maternally inherited, or they could occur somatically. So let's say during one's lifetime, right? There could be some sort of mutation that just spontaneously occurs. Yeah.
So maybe because of environmental factors, or
Right.
some I guess chemical imbalances, maybe, or what have you.
Right.
Okay, wow, that's very interesting. So yeah, I mean, at the end of the day, right, this is why we do what we do, right?
Right.
We actually can connect our work with solutions that could actually save lives, right? That have
Yeah.
real world physiological applications, right?
Right.
Yeah. So so why is this technology better than the current, I guess, mitochondrial DNA editing techniques we have now? Reading your work, you talked about zinc finger nuclease. So, I mean, is that a facilitator of what you're doing, or is that some other form of DNA, mitochondrial DNA editing?
Yeah. Okay. So I would say that our current technology is complementary to what is already out there.
Okay.
So, like as you mentioned, this zinc finger nucleases, right? So the zinc finger component is the DNA binding domain, the one that's responsible for finding the DNA. And the nucleases is the one that cuts the DNA. So it's the scissors that basically shears things up.
Okay, so the molecular shear, right?
That's right.
So kind of like CRISPR-associated proteins 9 or 13.
Right. That's right. So previously, what the toolbox that we have enabled us to cut mitochondrial DNA. So it's useful if you identify a mutation that this person has a mutation in a particular sequence that's deemed to be pathogenic, and then you could use this zinc finger nucleases to home in on that mutated DNA molecule and cut it and shear it, basically, to remove it. So you leave behind only like healthy copies of your mitochondrial DNA. Yeah. So what this deaminase, this new approach, what it does is that instead of cutting things up, you are changing. It's like a pencil where you change the DNA base. You erase and you change it. So you could make single nucleotide changes at your desired site. So in this case, our desired nucleotide change is a change from cytosine to thymine, a C to T change. And I guess this is useful if, instead of cutting things up, if you want to make specific nucleotide changes that they don't already exist in the molecule, right? So you can introduce new nucleotide changes or correct any mutations instead of just cutting DNA up. So we're not constrained by existing sequences in the mitochondrial DNA with this new technology. And I think the other advantage of this is that there are some cases where if the mutation load in the mitochondria is so high, you can imagine that if you decide to cut all the copies of your mutant DNA, you're going to end up with a cell with no mitochondrial DNA, and that's going to be harmful.
Yeah, we don't want that.
Yeah, we don't want that, right? Yeah, so this is particularly, so you cannot cut things up. You cannot use zinc finger nucleases when it's a case where the mutation load is so high. You have to find an alternative, and we are hoping that this method of making nucleotide changes instead of cutting things up could be the alternative for cases where the mutation load is very high.
Oh, that's super interesting. All right. So moving on. So the double-stranded DNA deaminase toxin A, which, by the way, that is an acronym that you guys actually came up with, right?
Yeah.
It's DDDA.
Yeah.
How do you actually pronounce that? Triple DA?
Yeah, it's just triple DA. Yeah, it's double-stranded DNA deaminase. It's a mouthful.
Okay. So what we're trying to do is to get that present in the organelle, right? So
right.
Now, can you, as simply as possible, explain how the base pair switch occurs, right? So
right.
you were just talking about it. So,
right.
for example, how do we switch from the CG to TA nucleotides, basically?
Right. Okay. So, so as you mentioned, deamination removes amino groups, right? So for cytosine, when you deaminate cytosine, you end up with a base that's uracil. And uracil has the base pairing properties of thymine. In other words, when other proteins see uracil, it's as though they saw thymine. So if it acts as thymine, you can imagine that during replication, where that uracil containing strand is being used as a template, polymerases or DNA replicating enzymes would incorporate an adenosine instead of a guanine when they seize the thymine. I mean, when they seize the uracil, sorry. So that's how, because uracil behaves like thymine, and that's how you get from a CG to a TA, because the intermediate uracil looks like a thymine to the cell.
So it sounds like genealogical espionage.
Right, yeah.
Okay. So now, what are tail array proteins, by the way, and how is combining them with your triple DA make for a relatively good gene editing, efficacy?
Right. Yeah. So these tail proteins, they're also DNA binding domains, like DNA binding proteins, and they are naturally occurring. So they are found in like pathogenic bacteria again
Oh, wow.
that target like certain genes in plant cells, right? So these are naturally occurring proteins, and they've been repurposed for gene editing applications because they can be reprogrammed to bind to different DNA sequences. And how they go about doing this is that, so for each tail protein, it's made up of many repeat regions. So each repeat region could be about 34 amino acids long. And you could imagine that this thing just repeats itself for how many times you need, depending on the length of the DNA that you want to recognize. So if you want to recognize, say, 10 base pairs, then you would have 10 repeats, each repeat responsible for recognizing one DNA nucleotide. And to specify that nucleotide recognition within this repeat, all you need to do is just change 2 amino acids at positions 13 and positions 14. And there are 4 possible amino acid changes you could make to correspond to the 4 different DNA nucleotides that you can recognize.
Wow, that's awesome. That's some very mind-bending stuff.
Yeah, it's pretty amazing.
It is amazing. Now, as we know, right, so
Right.
this is more a somber, I guess, topic. As we know, gene editing is not foolproof, right? So I
Right.
remember watching a talk by Professor Fan Zhang,
Right.
and he was talking about how Cas13, right, so CRISPR-associated protein 13, might be more favorable than Cas9 because I guess Cas13 edits RNA, which can then be reversed, right? So in case
Right.
wrong edits, right, RNA edits could be reversed.
Right.
Whereas Cas9, which edits DNA, those edits are basically permanent, right?
Right.
Now, what I want to know is, are there any, I guess, contingency plans in place in the case of bad edits, right? So with what
Right.
we're doing, or are bad edits not as, I guess, consequential in mitochondrial DNA editing compared to editing in the cell nucleus?
Right. Okay. So maybe I'll define like bad edits as unwanted edits that you make that can alter like protein function or maybe affect certain regulation, like gene regulation, for instance. And like you said, any bad edit, yeah. So any bad edits cannot be tolerated in any form of therapeutic. I think so to identify like these bad edits, right? In most cases, you would probably sequence the entire genome of the organism that you're editing, and to see where those bad edits are. So the approach is more of how do you prevent any of these bad edits from happening, especially since we're editing the DNA instead of the RNA. And that's when you have to go back to the drawing board and see, okay, should I improve the specificity of this DNA binding proteins to make sure that they don't end up at sites that we don't want? Or is there a way to control the activity of the deaminase such that you only turn it on for a certain period of time instead of leaving it active throughout the entire time where you administer the protein? So I think it's there's a lot of research that you need to make sure that you improve this, you create as specific editor as possible to minimize the amount of bad edits that you make into the mitochondrial DNA. And for your question of whether are they less consequential in the mitochondrial DNA compared to editing in the cell nucleus, there's some argument for it that yes, it might be true given that there are so many copies of the mitochondrial DNA. So let's say if your bad edit only happens maybe in 1% of the DNA population and the remaining 99% is healthy, that person should be fine. But I think this is a problem if when the bad edit actually confers some sort of fitness advantage to the cell. And you can imagine if it's only 1%, over time that 1% could increase to become 10%, 20%, and eventually overtake your population. So that's also not desirable. So I think the bottom line is, if it's used as a tool for like just disease modeling, right, within a controlled like in vitro tissue culture system, then it's probably okay. But if you're talking about editing of other organisms that are outside of the petri dish, then we really need to be careful and make sure that you minimize the bad edits to make sure that it's just at background level.
Interesting. So basically, the moral of the story is bad edits are deleterious no matter what sort of DNA you're editing, right?
That's right.
All right, that's very good to know.
Yeah.
The fact that you guys are thinking about this, and that makes me feel that there are certain contingency plans that are being considered, at least, right?
Right.
So now let's talk about the ethical ramifications of gene editing, right?
Right.
Now, being a researcher in this field, surely this brings you willingly or reluctantly, right? How you want to put it? It brings you close to the question of should we be doing this in the first place, right? Are we, quote unquote, playing God and so on, right?
Right.
Now, I know Professor Jennifer Doudna of Berkeley.
Right.
We're not gonna go into that.
Yeah, I know what you mean. Yep.
Anyone from Broad starts raising the eyebrows, right?
But then to clarify, actually, so my advisor has good working relationship with both people.
Oh,
yeah.
wow.
So we've not specifically collaborated with Feng's lab before.
Sure.
We definitely have had many ongoing collaborations with Doudna's lab.
Oh, wow.
Yeah.
So there really isn't like a conflict of interest, though, right? Because
you're not
necessarily working closely with Professor Fang Zhang, right?
Right. Yeah. So actually, it's quite nice to be in David's lab where it's okay with both sides.
Yeah, you guys are an intermediary between
the
warring factions.
Yep.
That's cool. What about Professor? Is it Emmanuel Charpentier?
All right. Yes. So I think we haven't had any collaborations with her yet, because I think she does a bit more of the basic biology side of things, which is equally important. But then our lab hasn't ventured that far into like her area of expertise yet.
Okay, interesting.
Yeah.
So, as I was saying, Professor Doudna thought out in the past a moratorium on which kind of gene edits, right, are morally justifiable, right?
Right.
So now, this hasn't stopped certain scientists from editing human embryos to possess certain, I guess, genealogical advantages, right? So this brings in mind the work of Chinese researcher. I think his name is Dr. He.
That's right.
He edited human embryos to have mutations on the CCR5 gene, and this was actually done in a set of twins, right? So it's from developing AIDS in the event they get the HIV virus, right?
Right.
Why he would pick this specific area, I have no idea.
Yeah.
But that's besides the point. Now, how dialed in is, I guess, your moral compass, if I may say, right?
Right.
On what's ethical, what's borderline, and what we should, I guess, totally avoid when it comes to gene editing.
Maybe I'll start off with what we should totally avoid, at least in my opinion.
Sure.
Right. It's like I'm on the side of the Professor Jennifer Doudna, where she proposed a moratorium and said that at least with what we have currently, any editing of eggs or like sperms or like germline editing with the intention of implanting it and producing like children.
All right. Should we actually explain what germline cells are? So so germ cells are basically cells that are not somatic cells, right? So every cell would be a somatic cell. So the reproductive cells would be our germline cells, right?
That's right.
Right.
And they are the ones that go on to like create offsprings, right?
Yeah.
So that's where I think for me, I don't think whatever we have right now is suitable for germline editing, with the intention of implanting it into um, implanting it and producing children.
Right. Yeah, because this could actually go down the germline, meaning it
Right.
can actually affect
Affects generations.
the progeny
Right.
of the individuals that are having their germlines edit.
Right. So, which is basically what the work of Dr. He did, and so that to me is something that we shouldn't be doing as of now, and especially since um, they don't have a good reason for doing so. So in that case, it was to protect children from HIV infection. But then there are other better methods out there that don't require editing of your germline. So it is really, really uncalled for, right? So germline editing is probably the big no-no for me right now. And as to like what can be done with gene editing, it's like, to me, I think crops is something that is something that's worth considering as a candidate for genome editing, because I know there are some concerns about genetically modified crops, but if you think about it, actually, right, how agriculture have progressed like over centuries, it is actually a form of evolution, right? But it's just taking place at a much longer time scale. So with our current tools, if we can enhance the fitness of certain crops to make them grow faster or to produce crops that have high nutritional value, you actually can produce like a lot more food for the growing population. And that is something that is, at least to me, it's worrying when you see the agriculture being placed under so much stress, right? And especially with climate change, crops are not growing as well. It's really something that I think genome the genome engineering field can play a big part in it. But of course, it has to work hand in hand with how um, uh, the the companies and the manufacturers educate the public about genetically modified crops. So that's one aspect which I think gene editing has a lot of potential, and I believe that it should be done more. So in terms of like editing in like other organisms, like non-human organisms, um, so obviously you create animal models to study certain diseases. So that is also like a useful um, application, right, for disease modeling purposes. And as of now, if we talk about therapies, there are ways that don't you can do therapies on like gene therapies on humans without any germline editing. So it's a case where, say, for a lot of blood related malignancies, the idea is, can you extract the stem cells from the individual and then perform your editing outside of the individual in a petri dish, right? So you could edit those cells. You do your sequencing of those cells to make sure there are no bad edits, and if they're healthy enough, you implant them back into the individual. And then this, to me, I think that technology itself is proving to be really promising. There are companies who have been starting like clinical trials on it already. So I think, and especially since these diseases, there's like currently no good cure for it. And many of these patients, even if they do take drugs, they have to be on drugs for their entire lifetime. And there's no promise that they would ever recover, or they wouldn't, or their disease wouldn't worsen. So if it's a case of like a last resort, and if you're sure that it performed in a very safe and controlled manner, I think gene editing still, there's still the option to choose gene editing. If you take into account other factors like cost and um, um, the fact that there are really no other alternative options.
Wow, very interesting. So what you're saying is gene editing should be seen as another tool in our arsenal to combat these pathologies, right? That
Right.
not just our agriculture, but even our lives and the lives of our livestock, our pets, and
Right.
just to be a well-rounded species with tools to make life a little less painful, right?
Right. Right.
Very interesting, very interesting. So Beverly, I can't thank you enough for this. To wrap up, so what are your plans for the future, right? So
Right.
you're, I guess, face deep into your work at Broad.
Yeah.
What should we expect from you in the future?
Yeah. So I mean, I'm studying my fifth year of graduate school now. I'm hoping to learn as much as I can from this lab. David has provided an amazing environment, and I know it might not sound believable, but I really do enjoy every single day that I have to go into that. Like it's fun just being around these amazing people.
Why wouldn't that be believable? Like you're editing scenes. Come on.
No, but no, but some people think that, oh, like grad school, you have to go in lab every day, even on
Yeah,
weekends.
the drudgery of getting
Yeah.
to school, yeah, it's there, but I mean, I guess it depends on how much you enjoy what you're doing, right?
Right, yeah. So I'm thankful to be in a place where I really enjoy what I'm doing, as well as the company of the people that I'm doing this work with. So, in terms of future plans, so I was thinking of probably looking into an industry job. I don't think I'm suited for academia, so I hope to continue my research journey. Maybe in an industry position, whether it's like a biopharma company, uh, or an agriculture related field. I think that would be something that I'm looking forward to, and hopefully will work out eventually.
Awesome. So, are you planning on staying in the States, or are you going back to Singapore? You going to Europe?
Yeah.
So,
So
where's the wind gonna blow,
yeah.
Beverly?
So I think for now, for at least for the next 5 years when I graduate, the wind has to go back to Singapore. I mean, my family is there, but also like I mentioned at the beginning of the podcast, so the government agency funds me for my studies, and I think and I have to go back, to serve out like a bond commitment with them for 5 years, and after that, we'll see what the options are. I mean, there's
Awesome.
so many things to choose from, so
I bet. Yeah, the future is bright, right?
I hope so.
Awesome. All right. So, Beverly, if anyone like me reads your paper and they find it, illuminating, and they want to contact you, are you open to emails or?
Oh yeah, definitely. Yeah, I think my email can be found on the group's webpage, and I'm happy to, if there's any comments, questions, or feedback, or even if it's not related to paper, if somebody's interested in what's going on in the lab, I'm also happy to put them into contact with the relevant people.
Awesome, awesome. Well, on that note, Beverly, I do appreciate your time. Thanks for coming on. And this was, I mean, gene editing is, fun as it is, but the fact that you're able to make it digestible for me and the listeners makes it even much better, much sweeter.
I really appreciate you having me here. It was a good experience for myself to be on this podcast too.
You take care now.
Yeah, you too.